Okay, so we're going to continue on with another aspect of translation. And the question at the top says, how do we get from a strand of messenger RNA to a protein? And then it says enter transfer RNA, also called tRNA. Remember, there's three types of RNA. There's messenger RNA, transfer RNA, and ribosomal RNA. So this one is our transfer RNA, and it's on the left-hand side here. And sometimes it looks like a clover or a T. So we're going to label this transfer RNA, there's two different boxes we have to label, and then we're going to describe some things that it can do. So first, at the top, there's a circle and it says MET in there. MET stands for methionine, which is an amino acid. There are about 20 different amino acids that these transfer RNAs can be carrying, and methionine is one of them. And then at the bottom, we have a series of 3 letters. And now with messenger RNA, we call it a codon, but if it's located on a transfer RNA, we call it an anticodon. Oh, and then, yeah, our box here in the middle, that's just pointing to the transfer RNA itself. But then here, this series of 3 bases is the anticodon. Okay, so now we're going to talk about what does it do. One of the things that transfer RNA can do is it binds a specific amino acid. So whatever anticodon is down here, there's certain ones that match to certain amino acids. So they always go together. The anticodon on transfer RNA can bind to the correct codon on the messenger RNA, and we'll see that in the process later, but they're going to match up. So if there was messenger RNA underneath here, it would pair up with an A, a U, and a G on the messenger RNA. And the transfer RNA is going to bring the amino acid to its place on the ribosome, and then it kind of plugs in with this anticodon to match up to the messenger RNA. The amino acid can then form a peptide bond with the next amino acid, and it starts building the polypeptide chain. So that's what goes on with our transfer RNA.